Students, good morning. Today we are going to discuss the classical pathway of complement system. Okay, so classical pathway of complement system. So we have in the complement system, I think you're familiar with the different pathways are involved in the complement pathway activations. So generally we have three different type of the pathways. They are the alternate pathway and classical pathway. One more is a lectin pathway. Today we're going to discuss the classical pathway system for the complement activation. This is the like the structure of the C1 complement system, other complement proteins. Okay, so now classical pathway it is can be activated by the other dangerous signals like the C-reactive proteins, viral proteins, apoptotic cells. So due to this, all this pathway will be activated. This is the antibody dependent. Sometimes it will be activated. So both in antibody independent also. This classical pathway it is generally like initiated by the two different type of the antibodies, either in the form of IgM, it's a pentamer, this IgG, it is another antibody. So due to this, either IgG or IgM mediated type of the classical pathway activations. This structure you can see here, this is the structure of the complement system, right? We have a like C1, it is a complement system one. We have a series of the complement system, like complement protein like C1, C2, C3. Like this, we have a so series of the complement proteins, they will activate so one by one and they will initiate the cascades of the reactions. Okay, so this part it is a CQ. So this so it has another this tail like structure is a C1R and C1S. So the C1 that is a complement protein one, the Q part is bound to the surface of the antigen. Then it will triggers the conformation change in the like tail part of the C1R and C2S. So like that, it will start activating the classical pathway. So as I said, this will be inactivated because of the antibodies either in the form of IgG or IgM mediated complex. They bound to the C1 complement system of the Q proteins. That leads the once the C1 it is activated, like the C1 of the Q protein it is activated. So once activated, then it triggers the activation of C1R and C1S. Okay, so how it will activate it, because the conformation changes in the C1R and C1R in turn it will activate the C1S. Okay, so then so this activated C1S, so it will activate the other series proteated kind of the cleavage proteins. They are the C4 and C2 complement proteins. Okay, once a C1 it is activated, C1S it is activated, it targets the cleaving of the C4 complement protein as well as the C2 complement protein. That leads to the formation of the C3 convertase. Okay, so which in turn, once a CT convertase it is activated, which in turn cleaves the C3 convertase into the C3A and C3B. Okay. So while C3B act on the recruiting the inflammatory cells and C3B bind to the other form of the complex, that is the C5 convertase. Once C5 convertase is activated, it instead initiate the formation of membrane complex attack. Usually we'll consider as a membrane attack complex, MAC. This leads to the lysis of the bacterial cell membrane, and it leads to the complete destroying of the whatever the pathogen that's present in the membrane. So now, so in the schematic view of the different pathways, like classical pathway, it is mediated by the antigen antibody complex. Like alternate pathway, it is a pathogen surface, and lectin pathway, otherwise as the mannose binding pathway. So these all this pathway it will activate the complement system, and it will recruit the final at the end with all this pathway activation. It leads to the recruitment of the inflammatory cells, opsonization, and killing the pathogens. So like inflammatory cells in the palm of they'll activate the TNL alpha, all the cells like tumor necrosis factors, interleukins, all this they will completely destroy the pathogen. Now we'll go to the so this as we discussed that this classical pathway it is activated due to the presence of the IgM antibodies. This will bind to the surface of the bacterial surface. Okay, so because this IgM it has a pentamer. This is just a picture of the antibodies IgM. So it will go and bind to the surface of the bacteria. So usually C1 bind to the single molecules of the IgM proteins, otherwise immunoglobulins, whereas IgG is bind to the bacterial surface. So the C1Q so bind to the two or more molecules of IgG, like IgG antibody molecules. So now we'll go to the how exactly this pathway like the leads like the schematic representation of understanding the activating the C1 as well as the C3, C4, C3 convertase and C5 until the membrane compress attack. How it will happens, we'll go to the pictures and we'll understand the exact pathway. Students, can you look here? So this is a target cell. So whatever the bacteria or pathogen it has entered into the our body. So during that times the this pathway is activated due to the presence of two type of the antibody, like either IgG or IgM. Already I have explained. So once the site IgG or IgM is bound to the surface of the bacterial cells, then it will activate the C1 complement, the complement proteins one. So we have discussed the structure of the C1 complement, right? So we have a Q, R, S. Once the Q it is activated, so the head part of the complement proteins, that is Q, it is activated, then it will activate the S protein as the like the other part of the proteins. They are the like tail part of the proteins. This, right? This is R. So once this is activated, okay, head part, it leads to the it will give the signals for the conformation change in the C1R as well as the C1S. Once the CNR it is activated, it will activate the C1S protein. Okay, the C1S protein further it will activate the other complementary proteins. So activated C1 proteins, it will cleave the C4. So C4 complement complement into the C4A and C4B. Okay, C4A it will diffuses, and like this it will cleave the another molecules of complement protein, that is a C2. So C2 it is cleaved in the form of C2A and C2B. This C2B it is diffuses, and so this part of the C4B as well as the C2A, they form the C3 convertase. Okay, so remaining the smaller subunit, otherwise you can consider the C4A it is just diffused, and this cleavage of C2A it will be attached to the C4 like 4B of the complement proteins, and C2B it is diffuses. This bond like C2A and the C4B, they will be considered as a C3 convertase. Once the C3 convertase it is present again, the C3 convertase it is undergo the cleavage. Okay, this cleavage it will activate the C5 convertase. Then again the C5 convertase, so it will activate the other subsequent like complement proteins. They are a C5, like C6, C7, C8. You can see, C6, C7, C8, and finally they form the like nine membrane complex attacks. These are they form the pores like structure. There is channel like structures in the membrane. So here all this one the pathogen it lyses here. The membrane become unstable. Like this, it end up with the opsonization. Now coming to the significance of the classical pathway. So classical pathway, it is there's a micro center into the body. The body responds by producing the antibody. The antibody against the antigen. They are in the form of IgG and IgM. They form the antigen antibody complexes. Okay, this will be help for the adhere to the microbes to the neutrophils and macrophages, and facilitates in the induction of the microbes by neutrophils and macrophages to the phagocytosis. It binds to the lysis of the microbes and augments the IgM-mediated cell cytotoxicity. So this is about the pathway for the entire pathway for the complement system activities. So in this table represent the entire proteins like complement proteins involved in the classical pathway. They are the C1, what we discussed the structure of the C1Q, R, S, and the remaining start with the C4 cleavage and C2 cleavage. The part of C4B and C2A, they combine to form a C3 convertase. Again, the C3 convert, again it will C3, it will going to cleave, and it leads to the activation of the C5 convertase. And the C5 convertase, it will activate the further signaling molecules. They are a C6, 7, 8, 9, and leads to the membrane complex attacks. Okay, this is the one type of the pathway in the classical pathway of the classical pathway of the complement system.